Yeah, thanks everyone for joining us today. I'm Patrick Murphy. I'm the market segment manager for transcriptomics at Oxford Nanopore Technologies. Over the next 10 minutes or so, I'll provide a really brief introduction into Nanopore sequencing platform and touch on why long reads are particularly powerful for RNA analysis. I'll also highlight two recent RNA-related product updates that we have, our improved cDNA sequencing protocol, as well as our new approach to enable multiplex direct RNA sequencing. So if you're not familiar with Oxford Nanopore, it's really helpful to take a look at how we sequence, right? So at the center of technology is actually going to be the nanopore itself. So a motor protein controls the movement of a DNA or an RNA molecule through the pore, and changes in the electrical signal that occur as as the strand moves through the pore can be measured and then translated into sequencing information through base calling. And this means that as the molecule is being read, it's being base called sort of live. And that means that the information about that sequence is available nearly immediately to the researcher. You don't need to wait till the end of the run to get a big bolus of data. And because the platform analyzes the molecule directly, it can currently sequence both DNA and RNA molecules. And this means in the context of DNA, we can run unamplified genomic DNA straight through the pore, which enables us then to capture not just the sequence, but also the native modifications such as methylation that might be directly on that molecule. For RNA, we can also do the same. We can run native RNA right through the pore and capture modifications. And in either case, DNA or RNA, of course, we can also use a PCR amplified product. Each nanopore operates independently, and depending on the throughput that you need, there's two different flow cell platforms. So there's the MinION, which has fewer channels, fewer pores, and a slightly lower output. The PromethION, which has a higher number of channels and pores, which has a higher output. And typically for oncology studies, the throughput of the PromethION flow cells is going to be more suitable. And we offer, as far as sequencers, the ability to run, we have a benchtop sequencer and a higher throughput sequencer, the ability to run 2 or 24 flow cells simultaneously. But importantly, individual flow cells positions can operate independently. So you don't need to wait for a large batch of samples before you can begin your experiment. You could have one sample running on a 24 space sequencer very easily. And it's also worth noting that while the MinION platform does have the lower throughput, it has excellent portability. And so here on the left-hand side is an example of the Mark 1D. And this supports the full MinION chip, so 512 nanopores, but it just fits directly in the palm of your hand, which makes it really quite accessible. So just some background on why long reads are valuable in transcriptomics. One of the central challenges is that one gene does not necessarily correspond to one transcript, right? So most genes can actually produce multiple transcript isoforms through a process of alternative splicing or through utilization of alternative transcript start sites. And with short read RNA sequencing, an individual read often covers only a small part of the transcript, and it might not even cover an exon-exon junction. So as a result, the same short read can frequently be compatible with several different transcript isoforms. And the complete transcript structure then will need to be reconstructed computationally from these many different separate fragments. And this can make it difficult then to determine which isoforms are actually present in your sample and to quantify the individual isoforms with good confidence. Long-read sequencing, on the other hand, allows complete or near complete transcripts to be captured in one individual read. So in this little schematic here, rather than having little fragmented reads that fall within different exons or maybe between different exon-exon junctions, we're going to get all of the exons and junctions combined in one read. And with Oxford Nanopore cDNA sequencing, a read can extend across multiple exons and splice junctions. Transcript reconstruction therefore becomes much more direct, or maybe even not even necessary. You may just have the full transcript there, and there's a lot less reliance on inference. It also makes read to transcript assignment less ambiguous, supports more confident isoform level quantification because we're not trying to guess where each read falls into, and also really enables a lot of information for novel isoform discovery because presumably we're going to have the whole transcript there. The same principles of the value of the long read apply to structural events such as gene fusions as well, because you can detect the fusion with short read, sure, but with a long read, we can capture the surrounding transcript context as well. So we can capture a lot more that's on either side of that fusion event. And long read RNA sequencing, therefore, can address many of the same expression and differential expression questions that you'd have with short read sequencing, but it's also going to provide much higher resolution into the transcript structure, information about splicing and fusion events. So if you've worked with Nanopore cDNA sequencing in the past, were some really major updates that we've made to our protocol that are now offering significantly longer reads. And so on the plot on the left, we can see our old protocol in gray, our new protocol here in blue. And what we see is about a twofold increase in median read length based on a standardized sample of UHRR. We go from approximately a 900 base pair read length to about 1,800 base pairs. And this increase is really important because the longer reads are going to provide better coverage across the entire transcript. And researchers then are going to be able to observe more splice junctions within the same molecule and a clearer view of the transcript structure. In the lower right here, we have an example with COL1A1. It shows the practical effects of these improvements. So it's a really long transcript, about 18 KB, and the reads generated with the updated protocol here in blue. We can see we're getting much better coverage, especially towards the 5' end, than we did with our previous protocol. And the workflow changes, while they gave us the greater length, they still maintained the high output of the Prometheans. We're getting about 70 million reads per flow cell. Keeps still with accessible input requirements of as little as 10 nanograms of poly-A RNA or 500 nanograms of total RNA, and maintains some of the chemistry advantages we have as well with really minimal internal priming for good data quality. One thing to note is, again, if you've already used these protocols, or used our cDNA protocol in the past, the new protocol actually does not change any of the Oxford Nanopore kit components. It changes strictly the third-party reagents. So it's really straightforward for existing users to adopt into this improved workflow. Or, if you haven't used long-read sequencing from Oxford Nanopore before for RNA, protocol remains just about as straightforward as it was before. The second update that we made very recently focuses on direct RNA sequencing. So as I mentioned at the beginning, direct RNA sequencing is quite unique because we're actually sequencing the native RNA molecule itself. It's going to pass right through that nanopore. And so there's no requirement to first convert the RNA into cDNA, which means that researchers can examine both the gene expression and transcript structure like they would with cDNA, so all the information from cDNA sequencing, but they're also going to retain information of the native RNA molecule. So that's going to mean RNA modifications, poly-A tail characteristics, and things like that. And the technology currently enables detection of up to 8 RNA modifications from the same sequencing experiment. So we're going to be getting that epitranscriptomic information without the need to run multiple different assays to get multiple different modifications. But historically, one of the challenges that we've heard is that people trying to run native RNA samples, it was pretty difficult to scale because before this particular kit that I'm talking about here came out, it was essentially one sample to one flow cell. The new kit that was just released a few months ago is our direct RNA barcoding kit. It offers 24 different barcodes and allows up to 24 different samples to be multiplexed together and run on a single flow cell. This can then, of course, substantially reduce the sequencing cost per sample, and it makes it possible to do much larger direct RNA studies. And just a little bit of data to show sort of the performance here. On the left, we've got a plot showing the expected, I should say the barcode balance. And so the barcode balance is quite strong between the different samples. So you can be confident that when you load multiple samples, the number of reads assigned to said sample would be around the same as what you would expect for the other samples. And on the right-hand side, we can see that the expected barcode versus observed barcode is quite tightly correlated. So there's very little cross-assignment between samples. You can be confident that the data that's assigned to said sample is from said sample. So with the release of the new sample barcoding capabilities for native RNA, native RNA sequencing now is more accessible, more scalable, and more cost-effective for researchers that are interested in understanding both the transcript expression and structure, as well as RNA modifications and/or poly-A tailing. So just to summarize, Oxford Nanopore RNA sequencing is designed to provide really a combination of scale, flexibility, and biological resolution. Long reads that we offer improve the capture of full-length transcripts, and they're going to provide a clear resolution of splice variation and isoform information. The ability to sequence complete transcripts is going to support just generally isoform resolution, but also is really powerful in novel isoform discovery, as well as still providing information on differential gene expression and differential transcript usage. The ability for Nanopore to sequence RNA directly adds another dimension as well by enabling researchers to examine RNA modification and poly-A tails while preserving their actual information directly in, so to say, the actual information and position from the actual transcript, original transcript. And these capabilities are supported by an end-to-end workflow covering RNA extraction, enrichment, library prep. We have all protocols for that, as well as downstream analysis through our software, the Epitome analysis platform. So, our overall objective is to make transcriptomic studies more complete, more scalable, and easier to adapt as the research questions continue to evolve and our understanding of biology and cancer biology also evolves. So with that, I'd like to thank you again for joining the webinar today. I'm now very happy to introduce Dr. Adam Shlien. Dr. Shlien is a senior scientist at Sick Kids Children's Hospital in Toronto, Canada, where his work has an emphasis on the analysis of pediatric cancer genomes using cutting-edge sequencing tools. Recently, Dr. Shlien and collaborators have introduced a novel diagnostic framework for cancer based on detailed understanding of their transcriptional uniqueness. And today he's going to present his research on rapid and accurate pan-cancer subtyping using RNA sequencing. So I will hand it over to Adam.
Thanks, Patrick. And thank you to the EACR and to Oxford Nanopore for hosting this session. I'm relatively new to Oxford Nanopore sequencing, but so I'm presenting some unpublished data from the past year or so, but it's been a lot of fun, and I think the technology in particular, I think for RNA-seq has a ton of promise. And so here are the disclosures from the company and ONT. My disclosures will come in a couple slides as well. So as we mentioned, I'm going to be talking about pan-cancer subtyping using RNA-seq. I'll tell you about the system that we established first off in terms of the algorithms, and then jump into the ONT specific bits. So here are my disclosures that the algorithms that are going to be discussed have been licensed to a startup company of which I'm a co-founder. And here's the challenge that we're facing is that still in high-income countries such as Canada, the US, and the UK, disease-associated death past infancy is really still dominated by cancer. And in a population of about 80 million, which is the population of Canada, we have about 4,300 new diagnoses of pediatric cancer per year. And in that, there are about 1,400 kids who will relapse or develop refractory disease or develop metastasis. And it's really at that point when the survival begins to drop tremendously. And that's true whether you're talking about brain cancers, sarcomas, solid cancers, or liquid cancers, leukemias. And for this reason, Sick Kids established the KiCS cancer sequencing program as a way of using genomics to uncover actionable mutations in these kids. This is a very busy slide representing a summary of the past 10 years or so of doing this kind of sequencing. I'll just highlight a couple things for this audience. One is there have been about 1,000 patients that have been enrolled. This slide is a little bit out of date. Most of the data that we've generated from both DNA and RNA has been short read, but I'll show you the potential of replacing a lot of that with long read data. We've done kind of the things that you'd expect, whole genome sequencing of tumor as well as matched normal, a comprehensive cancer panel of our own design, uncovering 864 important genes, as well as full transcriptome short read sequencing. And the bulk of this talk will talk about RNA-seq and its utility. But just generally, go back again, we find that across the whole cohort, that at least 60% of patients derive some kind of clinical benefit, or there's some clinical benefit from their sequencing. And a good 50 or so percent have clinically actionable variants that would informed on treatment. So that might mean putting them on a new clinical trial or administering a specific kind of therapy that would not otherwise be thought of. And really my job at SickKids is to continue to push this sort of flywheel forward, where this data and this benefit converts into new research projects, which allow us to then refine the assays to the benefit of the next generation of patients. And probably the most information-rich data that we've been looking at is from the RNA, not because the DNA, whether talking about the genome or the methylome, is not useful. It's just that the RNA provides a sort of live activity dashboard of how the tumor is responding to chemotherapy, which genes are being overexpressed in the present day. And we use the RNA-seq as a sort of platform to which we can detect fusion genes, internal tandem duplications, which are massively important, expression outliers, and for the subject of this talk, really talking about tumor classification, we're really using it as a potential diagnostic aid. And of course, other folks are using RNA-seq, both with short-read and long-read data, to really uncover the diagnosis of rare disease patients, and in particular, look for aberrant splicing defects. And so to this end, we developed two complementary algorithms, one called RACCOON, which is an unsupervised clustering algorithm that does things in a sort of hierarchical manner, where you take in a totally unstructured and large set of gene expression data, matrices, you do some low information removal, and you recursively iterate over these discovery of multiple different classes. And here's an example with our first and published version of the RACCOON Atlas with 13,000 samples as reference, where we end up with about 455 different classes of both tumor and normal expression clusters. And I should say that we've since updated this with a larger set of tumors especially, and find, of course, as you'd expect, even more subtypes of cancer. But this allows you to discover and create an atlas of human cancer from first principles, from the data itself. And because it doesn't rely on the initial labels of what the tumor was initially called, you can discover these things from the data itself. And what you're seeing here in the UMAP is just the topmost level, the 27 major subtypes of cancer, and these then divide and subdivide, again, in the sort of a tree of a hierarchy until you get to 455 very specific subtypes of cancer. And then we trained a set of neural networks that we called OTTER that allows you to take one patient's RNA and then match it to the atlas and provide a probability that this tumor matches to a high level, say as a neuroblastoma, and then the subtype within that neuroblastoma, with a sort of a probability that allows you to decide whether to use this classification or not. And we found that it's extremely accurate in both confirming or updating the original diagnoses. And here we're seeing sort of a fine wedge of this large graph pointing to a very specific subtype with a very high probability. It's robust to noise and sequencing variability, and I'll show you some data on paraffin where there certainly is a lot of noise and a lot of sequencing variability. And as you add more data, more stuff comes out. So we're continuously discovering more subtypes of cancer, including those that are clinically relevant for the prognosis of patients. And so in the original atlas of 13,000 specimens, we were able to uncover many of the major subtypes of liquid cancers, solid cancers, brain tumors. And here's just a slide showing you the ALLs, where you see tumors that are based on their major lineage, ALL or AML, but also subtypes that are defined by specific fusions, specific deletions, a specific overall expression profile called pH-like, and others. And so all of this really ends up giving you these kind of molecular definitions of each of the tumor types. And as I mentioned earlier, as you add more data, interesting and important prognostically relevant subtypes begin to emerge. So osteosarcoma, a bone cancer, which is primarily found in kids, divides across the whole atlas of tens of thousands of tumors into two major locations. And then in one of the locations, we see novel subtypes representing 5 or so different variations of osteosarcoma. And importantly, whether looking at overall survival or progression-free survival, we see significant differences. And these are differences that would not necessarily be apparent when looking at the osteosarcoma under the microscope. These are expression differences that define high levels of MYC alteration, as well as downstream targets of MYC and other aspects of the transcriptome that define this kind of worse, more aggressive version of osteosarcoma. So with David and Anita, as part of the KiCS Precision Medicine program, which I already introduced, we asked if this could be implemented clinically as a diagnostic aid. Can we begin to roll this into our usual standard? Every Tuesday afternoon, we get together and discuss with the oncologist, pathologist, and genomicists a good 2 or 3 cases from sick kids or others, and we make sure to, as much as possible, include the RNA-seq for us to be able to do this classification in real time. And what we have seen thus far, and part of our paper, is the confirmation or updated diagnosis for between 89 and 95% of these pediatric patients. So this includes 300 patients from sick kids, as well as 300 or so from our colleagues in Australia. And I'm just going to orient you to this graph because I'm going to show you a few other of these as we go through. This in the center is a probabilistic class assignment to a major area of the atlas. And then as you move towards the periphery, you're getting to more and more specific subtypes of cancer. And I would just say that probably within level 2 or level 3 is when the clinical utility of these subtypes stops. And then as you go beyond that, you get to really interesting research results. But I would say probably for the clinical use or clinical considerations of this platform in the future, one would think about levels 2 or 3. And this is already beginning to have an impact, though here is a patient of David's, in fact, who had a sarcoma NOS, which means we don't really know what the tumor is. And for that reason, they were given standard of care chemotherapy, and the tumor continued to grow. And this young boy of 10 years old really had this very large mass. I'm not a radiologist, but even I can see that there's something very big here. And we were able to take the RNA, run it through the classifier, and have it matched to a very specific subtype within the overall atlas that allowed us, together with our colleagues in pathology, to re-diagnose the cancer as this entity, so something a bit more specific, which had a change in the therapy and then an immediate response. And sure enough, this boy is back in school many years later and doing tremendously well. And there are a number of other examples like this where the original diagnosis is one thing, and as we apply the classifier, we get something that's a bit more refined or completely different. And Pedro, who's a ML specialist in the lab, recently came along and said, well, we should think about changing the way that we assign these probability scores. And I won't get into the huge technical details here, but essentially the way our published algorithm works is it allows equal odds for any of these 455 different classes to be assigned to the tumor of interest. And then Pedro said cleverly that we should use the major subtype, whether this is a brain tumor or an AML or an ALL, to then divide and guide the classifier as it goes forward and decides what the subtype and subtype is. And the end consequence of Pedro's change to the classifier is that now we are much more robust to these out of distribution samples. And so that means it doesn't necessarily have to be a human tumor. We can handle other cancers as well. It doesn't necessarily have to be fresh frozen. We can handle paraffin as well. It doesn't necessarily have to be poly-A selected. We can handle ribodepletion as well. And critically, as I'm about to show you, it also handles long read data quite well. And just as for folks who are interested in cancer research and preclinical models, this is just a plug. If you're running a model, whether it's an osteosarcoma, a rhabdomyosarcoma, or anything else, please check it using the atlas as a way of validating it and seeing that it actually recapitulates what we know about the human cancers. And if you're more informatically inclined, you can use our API, which more than 100 cancer groups have used to classify over 10,000 tumors. The website, well, I didn't put it over here, but I can put it in the chat as well as send it to you afterwards. All right, so now this is what you're here for, to see how this performs with a Nanopore RNA sequencing. And I think the benefits have been really beautifully outlined by Patrick. low upfront cost, easy setup, real-time data access. It's actually the real-time aspects of the Nanopore that's more exciting to me than even the long reads. And so we asked whether we could marry this informatics platform as well as the know-how and the atlas that we've developed over the years with the speed and portability of the Nanopore. And so Sandy, who's a bioinformatician in the team, started off by comparing matched short-read RNA-seq to long-read Nanopore sequenced data. And so here on this slide, you're seeing a number of different cell lines, some of which have been repeated, representing breast cancers, leukemias, and others, and the OTTER classification score. And you can see that by and large, it is correct with certain probability scores. Breast cancers go to breast cancers and onwards. And then when you do the same thing with ONT RNA-seq, in this case, using data from this publication, this was direct RNA sequencing, you see that things match the same. And in fact, in some cell lines where there's a second match to a different place, perhaps because of contamination, we see the exact same thing in both the short read and the long read. So this gave us an initial hint that we could use our atlas as it is, trained on short read data, but then deploy it for using this new and kind of immediately real-time available long read data from Nanopore, which was super exciting. And then what Sandy did was to ask the question of how much data do we actually need from the Nanopore to be able to classify our cancers correctly. And she did a number of experiments. Here, the major classification score or the subtype is depicted by this blue color, which matches the blue color at the top. And so what you're looking for is the emergence of this light blue color and the number of reads that are necessary for that to begin to emerge. And you can see that, really with perhaps 500,000 reads, and in some cases, probably a lot less, you can begin to classify the cancer. So for instance, here, just to drive the point home, in leukemia, you see this red very early, which means that with a subset, small amount of data, you could have classified it quite well. And I'll show you more about that as we go. And so seeing that our algorithms were compatible with the Nanopore without necessarily needing to retrain anything, we began to draw on a larger cohort of 174 tumors, including leukemias, solid tumors, and brain tumors, and at least a couple dozen tumors that have known gene fusions. And of course, gene fusions are massively important in pediatric cancer in terms of helping with the diagnosis of the cancers, as well as directing some specific therapeutic choices. We did both direct RNA sequencing, cDNA, PCR barcoded RNA sequencing, as well as the new multiplex version of the direct RNA sequencing. We also did a number of samples, put them through the Nanopore DNA sequencing, looking at methylation analysis. That's still ongoing, both by shallow as well as deep whole genome sequencing, and put it through the exact same classifier using the same workflow as I already presented. And so here's a screenshot of an expected fusion gene, and this is in a Ewing sarcoma. Ewings are defined by this, what we call pathognomonic fusion between chromosome 22 and chromosome 11, EWS and FLY1. You can see the evidence for this fusion, both in the direct RNA as well as the cDNA. And I can say that across the whole cohort of more than 20 tumors, 100% of them with gene fusions captured by the short read data were also captured by the long read. So really good. And what we see as we analyze the whole cohort together is over 90% concordance for paired samples where we have both the long read and the short read, and extremely high concordance for the clinical diagnosis of the brain tumors as well. Of course, many of those have been subtyped into different groupings of medulloblastomas and others, and we're able to find that as well here. And here, just to show you the paired plots that are constructed in the same way that I already presented here, the Oxford Nanopore data, cDNA is shown on the left in the short read, and I just want you to appreciate that even down to, let's say, 1, 2, 3, 4, 5, 6 levels deep, you have identical classification for this infantile fibrosarcoma. alveolar rhabdomyosarcoma, another alveolar rhabdo, an embryonal rhabdomyosarcoma, synovial sarcoma driven by fusion, as well as some other fusion-driven cancers that I've presented, an AML. And here, what we began to do is to use the barcoded direct RNA that Patrick presented as well. And we're just analyzing some of the first data from that kind of as we speak. But so far, a lot of the classification scores look great. And so here we have a triplet of the direct RNA sequencing on the left, the Oxford Nanopore cDNA RNA sequencing in the middle, and the short read poly-A RNA sequencing here. And there's a little bit of subtle difference, but as I mentioned earlier, the major clinical subtypes of the disease, which in this case is the diffuse high-grade glioma, are maintained across, and we're super excited about the direct RNA sequencing barcoded edition going forward. So then Sandy asked is what happens when you retrain the short read classifier, when you begin to add more data to it, what happens to the ONT data? So Ewing sarcoma is an important tumor type that's studied in the lab. And you can see initially that the Ewingsness, which is shown in this kind of pukish green color, emerges after 500,000 reads. And that's with a handful of less than 100 Ewing sarcomas in the training short read data. What we then did was added 600 new Ewing's tumors into that, retrained the classifier on the short read data, and then remapped our long read Ewing sarcoma tumors to it. And what you see strikingly is that this color of Ewingsness begins to emerge much earlier. And then in fact, you can diagnose the Ewings with only 5,000 reads, which is a fraction of a percentage, as opposed to the 500,000 reads that you would need to be confident before. So what this means is that as we add more data to the short read platform, to the atlas, as we improve the atlas, as we add more samples, it becomes not only faster, but also less expensive to classify new tumors going forward. And then just to drive that point home further, this is for a subset of our cohort. We analyze the number of reads and the classification accuracy. And in our situation, we have scores for the major subtype, minor, and then subtype, which is 1, 2, 3 shown over here. And what we can see is that within 10 to 20 minutes of the data hitting the hard drives via these POD5 files that ONT makes available, we get a disease group being revealed. And then within perhaps about 2 hours, we get extremely high confident classifications as well as a fusion gene being detected. And so in the next 5 or 7 minutes, I'll talk about a collaboration that I had with a colleague, Alan Zhao, in Kenya, as well as some case examples here at SickKids. So I had the really great fortune of being in Nairobi Hospital last year, in Aga Khan University Hospital, hosted by Alan as part of a two-brief sabbatical last year. And in Kenya, the survival of childhood ALL is really quite low, between 20 and 30%. And a lot of the challenge is the absence of diagnostic clarity. And so for this, in collaboration with Alan, we began to investigate using the ONT platform, using specifically the MinION that was available at AKU in Nairobi. We started by containerizing a lot of the bioinformatics workflow, which is available and people can use both for Nanopore as well as non-Nanopore purposes. We optimized the library prep protocol, although now with the new release that's been described, this isn't necessary anymore. In fact, the new release does give us a ton more data. And with Stanley, Lewis, and Matt, we began to look at the results as well as ensuring that the atlas had the relevant subtypes of ALL as well as TLL represented. And the results, which continue to flow in. So there are between 10 and 15 cases that have already been sequenced by Alan's team. And we can see that they really match the standard of care, which is flow cytometry. but they also allow you to go a little bit deeper and unveil things that are not necessarily easily available in that context. For instance, subtypes of BALL with a pH-like phenotype, AMLs that are driven by KMT2A rearrangements, and others, all with the same data and using sort of the power of the RNA-seq coupled with the informatics platform that we created. And then here locally at SickKids, we began to deploy this for ongoing patients, in particular leukemia patients, but also some solid tumors. And just to show you a couple examples, here is a patient enrolled in our precision medicine program, a young child with a TALL and a particular signature. By DNA analysis of our cancer panel, we found their TMB to be not elevated. They did have specific actionable variants that are shown over here. And what the oncologist really wanted to know was whether they had a specific subtype based on LCK overactivation. And sure enough, they mapped quite beautifully and cleanly to this GATA3 enriched subtype. And LCK was amongst the top 5% of the expression genes, leading to a suggestion of using dasatinib for this patient going forward. All delivered via long-read sequencing extremely rapidly. Another example of the utility of this rapid diagnostic aid is this infant with a tumor growing on their heart. The tumor is called an IMT and often has these fusion genes involving a partner called ALK or ALK. If one has an ALK gene fusion, then they might be eligible for crizotinib or some of the variations of those drugs. But when the sample came to us, no ALK fusion was able to be detected by standard of care pathology, which included a very deep dive and analysis by NGS. And strikingly, within 4 days of having received the RNA, we're able to find clear evidence for a very cryptic but important CLTC ALK fusion. And within 2 days after that, the patient was put on a clinical trial involving an ALK inhibitor and has really responded. And the net benefit, of course, will be seen in the long run, but for now has meant that they can avoid a heart transplant entirely. And we think we could have gotten this result a lot faster. We're beginning to become a little bit more organized so that we can do exactly that, as well as part of a Azrieli Foundation funded partnership with St. Justine Hospital. We began to put all the informatics as well as the protocols in line to be able to do this quicker. And you can see if you retrospectively look at the data that the dominant fusion really appeared after maybe a couple hours and began to be quite clear after 6 hours. So we probably could have reported this out even faster, and that's what we'll be doing for ongoing patients. And so I think I'll end here a couple minutes early even to show you that I've tried to describe an RNA platform for cancer diagnostics. It has high accuracy, even for rare tumor types. It's ever learning. New subtypes are beginning to emerge. Many of them have prognostic relevance and some have therapeutic relevance as well. It works with paraffin single cell line models and now with Nanopore data. And the addition of the Nanopore, I think, is hugely exciting for the potential of us having this distributed testing where people sequence locally and then use a common framework to analyze and potentially to diagnose their tumors. And then just to thank the team. I've mentioned the people already by name. So I think we can move over to the questions.